Good day, good day, good day. We are just waiting for a few more people to join in, and we'll start in the next minute. Okay, people are still coming in, but we'll start. Good day, everybody. My name is Mela. I am from Rare Diseases South Africa. I'm a community manager and also manage our memberships as well within the organization. We are joined by two beautiful ladies, Mel and Nabila, today, who are our guest speakers. I just wanted to do a few housekeeping rules. If you'd like to unmute yourself or say anything, please raise your hand. If you have any questions during the course of the webinar, you are more than welcome to leave your questions under our Q&A tab, which should be positioned right at the bottom of your navigation bar. If you are experiencing any technical issues, you're more than welcome to also write your query or grievance on our chat button as well, which is right next to our Q&A tab. So welcome, everybody. I would love to introduce our first guest speaker for the day, Mel. Hi. So Dr. Mel is COIN's unit director / a clinician scientist as well. Melissa is the director of the multidisciplinary COIN unit at the University of Cape Town's Neuroscience Institute. With a background in clinical medicine, genomics, and bioinformatics, her research focuses on bridging research and real-world healthcare. She's particularly committed to building a collaborative, locally relevant genomic capacity and advancing precision medicine for patients across Africa. Whew, that's a mouthful. Welcome, Doctor. The floor is all yours. I have a confession. I know nothing about healthcare professions or neuroscience, so I'll be learning as well. I will have questions as well, just to understand further as well for our community. Take it away, Doctor.
Thanks, Mila. Could I ask you perhaps just to introduce Nabila, because our talks will flow. They're just the same thing, so we don't have to stop midway, and then I'll get going.
That's not a problem. We are also joined by our lovely Nabila, a good friend of the Rare Diseases South Africa organization. So Nabila is also part of the COIN unit. She is a genetic counselor for variant scientists as well. She supports patients and families to understand the genomic results and what participation in research means in African context. Her work includes curating genomic variants using both global frameworks and local population insights. She facilitates responsible data sharing and through research contributing to healthcare systems that center people as much as science. So that is our lovely Nabila as well.
Thanks, Mila, and thank you, Rare Diseases, for offering us this platform to share our experience in using long-read genome sequencing to diagnose individuals with rare diseases in South Africa. And we'd like to use this presentation to share what we have learned so far in using this new technology in our research practice. So to start off with, Coin is firmly invested in genomic medicine and in the growth of this discipline in South Africa. And essentially, when we speak about genomic medicine, this involves using a person's genomic information as part of their clinical care. Coin's particular focus is in using genomic sequence data for diagnosis and specifically for rare diseases that fall in the neurology domain. So I wanted to ground this talk and our experience in a particular group of disorders, which I will refer to as inherited neuromuscular disorders, and to showcase how we have been using genome sequencing to attempt to identify the genetic cause of these conditions. So this group of disorders is a heterogeneous or mixed group of rare diseases that involve the brain, the muscles, the motor neurons, the peripheral nerves, or the neuromuscular junction. They can be present from birth to late adult onset, and many of them are progressive and without treatment. So inherited neuromuscular disorders have diverse genetics, and what we mean by this is that many different genes can be involved in producing these phenotypes. And in addition to the many different genes, there are also many different kinds of variants in these genes that can cause this broad group of disorders. Some of the clinical phenotypes are recognizable, and certain genes can be suspected on a clinical basis, while others are much more difficult to pinpoint particular gene candidates. So I'd like to start with the backstory of our experience being involved in research consortia as largely a genome analysis and bioinformatics group. So we were exposed to and participated in 2 international capacity building partnerships through our affiliations with neurologists at Groote Hospital. And these 2 consortia called CREATE and the ICG-NMD, which was involved with, well, basically it's called the International Center for Genomic Medicine Neuromuscular Disorders, ran for 5 years from 2019 to 2023. And they covered a broad array of inherited neuromuscular disorders. And the major goal for the ICG-NMD specifically was to partner with clinical sites in Africa and other lower middle income countries. And in doing this, increase the number of patients with neuromuscular disorders who could obtain a precise genetic diagnosis, which would then enable the delivery of personalized disease management plans and later access to clinical trials. So what were the findings? I think it's useful to anchor the talk in like a real world patient scenario. And for this, I'm going to speak specifically about participants who were recruited into the ICG-NMD consortium study from the adult neurology division at Groote Hospital. And as I said, the inclusion criteria were very broad, spanning all different kinds of inherited neuromuscular disorders. But I'd like to draw on 2 specific clinical cohorts to anchor the discussion. The first is a cohort of individuals with an inherited neuropathy phenotype, and the second, comprising 52 participants, and the second is a larger group of individuals with inherited muscle diseases, various kinds of diseases, comprising 89 probands. So what I've summarized here are basically the genetic findings. So looking at the proportion of individuals in these 2 cohorts who were able to obtain a genetic diagnosis through this research study. And overlaid on that, I've used the colors to indicate where the genetic diagnosis was obtained in country versus where it was obtained out of country. So participants could undergo local genetic testing if it was appropriate and available in country, and those where it was either negative or not indicated were offered whole exome sequencing, which was performed out of country in the UK. So if we look at the inherited neuropathy cohort of the 52 adults who were recruited over those 5 years, we can see that around half of them achieved a genetic diagnosis, and the local testing largely comprised evaluation of the PMP22 locus for deletions and duplications, which is a very common cause of inherited neuropathy, while the remainder of the diagnoses were largely achieved through whole exome sequencing out of country. And for these individuals who obtained a diagnosis out of country, what I've referred to or called this group is to say, in other words, that these individuals highlight a local genomic testing gap. So where genetic diagnosis was possible, but it was not achievable in South Africa. And if we look at this, a similar evaluation of the cohort with inherited muscle disease, where there were 89 adults recruited over 5 years, we see a somewhat similar picture. So overall, the diagnostic yield, meaning the number of individuals who obtained a genetic diagnosis, was higher. But we still had a considerable proportion of these, 52%, where the genetic diagnosis was obtained out of country, and the largest contributor to these diagnoses was, again, whole exome sequencing. In both cohorts, if I flash back to the other one, you can see that we still have a large proportion of individuals, about half in this cohort and about a third in this cohort, in whom a genetic diagnosis was not identified using the testing strategies employed in that research study. So where to from here? I've spoken about our involvement that we had in learning from our participation in these research consortia in terms of genome analysis and bioinformatics. And really what the COIN unit is trying to do in our efforts going forward is to try and build on the capacity that we gained from the consortium participation and really build on this in a local context. In order to do this, we have launched a new study called NeuroSeq, which is really a multidisciplinary effort involving ourselves as well as our clinical partners. And the flag here is to show you that the entire design of this multidisciplinary team research study is that the patients will be local patients, the genome sequencing will all be done in country, and all the analysis and interpretation will similarly happen within South Africa, so that we can invest in and really grow our genomic medicine infrastructure and ecosystem. And our hope is that this study will provide a large body of local evidence for neuromuscular disorders, and specifically the genetics of this group of conditions, which can really be used to inform research translation in this field. So just to really highlight our major goals with the NeuroSeq study, so the goals are twofold. The first is to try and close the local diagnostic gap, and on the other hand, expand the diagnostic frontier. So for the first aim, what we are trying to achieve is to offer equitable access to genetic diagnosis for all individuals with inherited neuromuscular disorders in whom a molecular diagnosis is currently possible, but to deliver this locally, sustainably, and in accordance with international standards. And then for those proportion of participants who are enrolled but who remain unsolved, through this study, we will actively pursue discovery science and cutting-edge sequencing technologies coupled with advanced bioinformatics and collaborative research to try and redefine the boundaries of what can be diagnosed. So in the first instance, for our first goal of addressing the local genomics testing gap for inherited neuromuscular disorders, the first area we tackled was whole exome sequencing, and I've highlighted it again here in the two cohorts to show you that by moving this sequencing technology and implementing it in country, we're able to really go a long way in improving the diagnostic yield. So 37% of the diagnoses in both cohorts were attributable to whole exome sequencing. So our tiered strategy for genetic testing is that where local targeted testing is negative or not indicated based on the patient's phenotype, we then want to move those analyses to whole exome sequencing, which we are conducting at the SAMRC Genomic Center. So just to touch briefly on the advantages of WEARS and why we have focused specifically on this technology. So whole exome sequencing focuses on the high yield part of the genome. So this is the roughly 1% of the genome which contains the protein coding regions. And these regions are known to harbor about 85% of the known disease-causing variants. As I showed you in the previous slide, we've already demonstrated a good diagnostic yield for inherited neuromuscular disorders using this technology at 37%. It is cost-effective. It requires less sequencing because you're only sequencing a small amount of the genome, and consequently, you generate less data which you need to store and analyze. And there is a low interpretation burden because focusing on the coding regions is where we have really well-established variant curation and interpretation frameworks. So it's relatively simple to tackle. To this end, we've developed a full end-to-end bioinformatics pipeline to process this exome data from the SAMRC. It gets delivered to us directly on our institutional high-performance computing cluster called ELIFU. And one of our bioinformaticians has developed a full end-to-end workflow to process this data so that in less than 24 hours, we can go from the raw data to the identified pathogenic variants. So to touch on the second aim, what I've spoken about now was our effort to close the local genetic testing gap. But what are we thinking about in terms of expanding the diagnostic frontier? So this would then be focused on this group of individuals in whom a genetic diagnosis was not achieved. And you can see that there are still a considerable proportion of individuals in whom this is not possible. So what we have to ask ourselves in this group is what are we missing and why are we not able to find the disease-causing answers? And so I've broken it down into a number of topics to discuss. So the first issue to consider is that this could be purely a genome space issue in that the whole exome sequencing here is focusing on the coding regions and there's a whole non-coding genome that we aren't evaluating. The second issue is the variant spectrum. So we know that through whole exome sequencing, we are really only targeting short variants that we can see on account of the technology using short reads. And so there's a whole spectrum of other kinds of genetic variants like structural variants and repeat expansions that we're not able to detect if we use whole exome sequencing alone. There might also be additional diagnoses that we can't conclusively prove without being able to phase them in the case of compound heterozygous variants for recessive disorders. And then we also know that there are short read NGS, so-called blind spots, and these are regions of the genome that are really inaccessible to short read technology like exome sequencing. And some of their characteristics include being GC rich, being repetitive, or having high homology regions. There's also the possibility that the genetic variants changes that we're looking for are not actually in the nucleotide sequence themselves, but maybe on the epigenetic layer or the methylation. So there may be other mechanisms that we're simply not probing when we focus just on whole exome sequencing alone. So in order to push forward on the diagnostic frontier and to try and really ask ourselves what we can do to try and solve these unsolved cases, we are using high fidelity long read whole genome sequencing at 30x coverage using PacBio's Revio platform. And this very advanced sequencing instrument is housed at Imkaba Biotech. And we ship our samples to Pretoria in order to access this sequencing technology. So what are the advantages in a nutshell of using PacBio's HiFi genome sequencing? So these are spelled out really nicely in the little pink blocks. So in addition to the small variants that we'll get, there are additional advantages, and these include the ability to detect larger structural variants, to profile tandem repeats, to look at segmentally duplicated regions, to phase our variants, to do advanced CNV calling, and also to look at the methylation. So it really is a whole suite of different kinds of genetic variants that we haven't been able to see before using short reads. Similar to our short read pipeline, we've implemented an end-to-end bioinformatics workflow to handle these long reads. And again, these get delivered to us from Incraba as the service provider directly to our high-performance computing cluster, Ilifu, where another one of our bioinformaticians has led the development of this workflow, where we're able to process the raw data to results in 48 hours, looking at a very comprehensive evaluation of all those different kinds of variants that I showed you in the previous slide. So really, this is to show you one of the very first views that we all gathered around the screen in our unit to look at, which really represents for us the dawn of a new era, and seeing what a HiFi genome looks like. And what I've shown you here in the two panels is the same research participant in whom we'd first performed whole exome sequencing, which did not lead to a genetic diagnosis, and in whom we subsequently did a HiFi genome, and showing you how these two technologies stack up against each other. And really in this slide, it highlights the additional genome space that you're covering. So if we look at this graphical representation of the gene at the bottom here, we see the colored in blue blocks represent the exons, and our whole exome sequencing really only gives us a small window onto the genome by covering the coding regions or exons of the gene, with virtually no coverage or no meaningful coverage in the intervening intronic regions. And you can see that in the HiFi genome, we get this complete unbiased coverage of the whole genome. But really the most standout feature between the two technologies is the difference in read length. So while the whole exome sequencing has read lengths of 150 base pairs, the HiFi genome sequencing can have read lengths of up to 25 kb. And I've just highlighted one of these reads here to show you that it's almost spanning an entire gene. And this really opens up a whole new avenue of opportunity when it comes to variant calling. So what I thought I would do next is to go through these, what I explained before, which are the particular advantages of long-read genome sequencing, and show you what we have discovered in our experience when analyzing HiFi genomes for the diagnosis of neuromuscular disorders, and showcasing the capabilities that we've uncovered with a particular focus on neuromuscular disorder disease genes. So the first major advantage is our ability to detect structural variants directly. And what I'm showing you here is again a comparison of the same individual sequenced with short reads and then sequenced with long reads. And what we can see, while we may have had some inkling by looking at the coverage drop here, but nothing conclusive, we can see clearly when you align the long reads to that region that we have a very clear heterozygous deletion of 837 base pairs. So we're able to pick up these large structural variants like insertions or deletions. The second major advantage, and particularly relevant for us because we study neurological disorders and we know that these tandem repeats or repeat expansion disorders are overrepresented with neurological phenotypes, is the ability to determine their sequence and repeat length and also the methylation. So what I've shown here is our detection of a CNBP repeat expansion, which is associated with myotonic dystrophy type 2. And I'm showing two things here. Firstly, our ability to accurately size the repeat at just over 230 repeat units, as well as the sequence composition. So we are able to clearly see, if you look at the repeating motif color-coded in the different colors, we're able to determine exactly what the composition is of that expanded repeat. Then moving on to segmental duplications, which is another group or another gene area where HiFi sequencing really excels. And for this, I'm showing you the well-known SMN1, SMN2 region, which is associated with spinal muscular atrophy. And basically, the SMN1 and the SMN2 gene regions are highly similar. And so when you try and sequence these regions with short reads, you have a problem because the 150 base pair length is not specific enough. And so when you try to map these reads back to a reference genome, you're not sure whether they actually originated from the SMN1 gene or the SMN2 gene. What subsequently happens, as I'm showing you on the top panel, is that while reads appear to map to the SMN1 region, the fact that they are white indicates that they have a very low mapping quality and essentially are rendered useless when it comes to variant calling. So this is demonstrating one of the short read NGS blind spots, which when you look at the long reads, because they're longer, are able to map more definitively to these two highly similar gene regions, and you are now able to get coverage of such regions. The next aspect that we've been able to determine is phasing, and what I'm showing you here is clear evidence of a transconfiguration of two variants. So the bottom panel is the HiFi genome, which is split into the two haplotypes, and here you can see that we demonstrate a compound heterozygous or transconfiguration of these two variants in the MPV17 gene, which have been shown to be a novel genetic cause of neuropathy among Black South African patients. And although you can detect the two variants in the short reads, you're not able to confidently say that they are on different alleles. And this can be a very important piece in the diagnostic puzzle. Next, moving to copy number variants. So our HiFi genomes and the companion software is very good at performing advanced calling of copy number variants. And to showcase this capability, I've looked here at the nebulin gene. This is a gene that's of interest to us because variants in this large gene have been shown or are known to cause myopathy, both in autosomal dominant and autosomal recessive forms. And what's particularly tricky is the central region of the gene, which is a triplicate repeat region. So it has multiple sections of the same sequence. And you can see if you look at both an exome and a genome using short reads that you essentially have another blind spot in the middle of this gene. And this is really important to understand or to cover because CNVs in this region have been associated with disease. And what we can see using the HiFi reads is that in addition just to the primary mapping, there's a companion tool called Paraphase, which essentially enables us to deconvolute all the sequencing reads into the different alleles. And we're able to show that there are 3 copies of the triplicate region on allele 1 and 3 on allele 2. So it's incredibly useful in evaluating disease genes. The last thing I wanted to touch on is methylation. So this is a whole another kind of variation that is kind of opened up to us to explore using long-read sequencing data. And we've done some preliminary investigation of the FSHD locus, looking at the epigenetic status of the D4Z4 array on chromosome 4. And what I'm showing you here is color-coded in red and blue. So blue shows us unmethylated regions. And so what we're able to detect is a contracted D4Z4 array, and also to have a look at the methylation status of that, which is an important diagnostic criterion when you're evaluating this locus, and is consistent with the permissive haplotype. So we're very excited about the ability to explore methylation directly in our HiFi genomes without having to do any other additional testing. So I wanted to tie this this up with this slide, which is a really forward-thinking uh, yeah, kind of topic, which is thinking ahead to a single genomic test for neuromuscular disorders. And what I wanted to, I've already highlighted that, we've brought whole exome sequencing in country. But when I scrutinized the remaining diagnoses that were achieved, and particularly those that were achieved out of country, one starts, if you look at them collectively, to see that they comprise repeat expansion, structural variants, and segmental duplications. And in fact, that's typically why they require a specialized assay, either MLPA or a repeat prime PCR, to detect them. And so if you consider that they all fall into these classes of variants, then we see that HiFi genome sequencing should really be good enough to detect them. And so if we then think about those remaining tests that we were not able to do in country, and we think about the potential of a single in-country HiFi genome, what we're really moving towards is an ability to reduce the local genomic testing gap to zero. As even though I haven't shown you concrete examples of all of these, by virtue of the fact of what kind of variant they are, a HiFi genome should be able to detect them in theory. And so we as a research community are finding this to be a very exciting prospect, particularly given the fact that HiFi genome sequencing is reducing in costs all the time. Even in the short time that we've been doing it, we've seen huge changes in the sequencing chemistry, which have translated into real cost reductions that start to make it more feasible. So I'm now going to hand over to Nabila.
Thank you, Melissa. The technology does bring much curiosity and excitement. However, for the patient, the cycle of testing from whole exome sequencing to whole genome sequencing, if unsolved, and then to long-read sequencing, adds another loop to an already long journey where, from symptom onset, they have undergone years and years of searching, living without answers, through many different stages of their life, to now having to mentally prepare themselves and adjust their expectations for another round of reinvestigating, which on their part requires a certain amount of reinvesting into this process, even though the outcome may take a long time and still may be uncertain. Parents are carrying a huge load, from fear to accumulated hope, but also accumulated disappointment by the time we see them for testing. So from a genetic counseling aspect, we need to recognize the emotional escalation from their side and counsel patients to cope for whatever the result may be without losing complete hope. I often get the questions of, is this the last straw, or this has to give us an answer? And I find that speaking to them more about why we're trying this new test now and what further information it can provide us, as Melissa spoke us through earlier, that even a negative result is also informative, and therefore it's still valuable because it rules out genetic categories and narrow down the future genetic tests that could be possible in the future. And from our experience, many patients do understand that they may not get any benefit out of it, but their findings can help understand and hopefully benefit someone else in the future. Next slide, please. This comes from the pre-test counseling and the informed consent, which needs to look more like preparation for the answers that they may or may not get, and not only asking for permission. As much as the consent process is procedural, it's also a way of empowering patients to make a meaningful choice for themselves and have a sensitive discussion with their family. We also need to prepare patients for future interpretive changes and, when possible, recontact. And as it's done in research, that their treatment journey and their clinical course may not change. Melissa, next slide. Because we enroll trials with long-read sequencing, hopefully the mom and dad, the process is a bit more complex as everyone in the family has a different experience navigating the condition and different emotions that they're holding for their loved ones. They sometimes also fatigued from the journey of testing and sometimes protective over why there is a need for another test. Some cases, their personal relationships are undergoing different strains that we only cover later on. So we need to facilitate a shared understanding and inform each individual, even though they at a different stage of readiness. It's important that they're seen as partners, but know that they have their own autonomy. The conversation around the return of results also is something that needs to occur beforehand. Is it going to be returned to individuals alone or as a family unit? And if the proband is unavailable or not in a state to receive the results, do we have the permission to deliver it to the next of kin? And apart from the emotions that I mentioned earlier on for the condition, with families, there may be guilt that they're carrying and blame, which we also need to be ready to meet and to unpack. Next slide. So I quite like this image created by Rhodas, highlighting the road through a diagnosis. There's many twists, turns, and ongoing cycles, but these quotes where a patient has said it is a waiting game, but you tell a mom to wait when she's waited for 15 years, it's very difficult. Or another patient that said that they went around traveling across the entire city to find a nursery for their son. It was impossible to have him accepted. And this just goes to show that how much of life is dependent behind the hope for a diagnosis. Next slide. We'd like to say thank you, especially to our extended COIN team. I do see some of your names online, and to our funders and collaborators as well. I will now hand over to Erin, who will share with you some ways that we can all positively contribute to the journey.
Hello, everyone. Thanks, Melissa. Thanks, Nabila. That was really great. So first on the slides that I want to just announce quickly is that COIN is coming out with a podcast in the next couple of months. This is something we're super, super excited about, and we're really keen for everyone to come and listen. The podcast is going to focus on genomic medicine in South Africa and what the landscape is currently looking like. And we're really excited for all of the guests that we've invited onto the podcast. And we think it's going to be a really, really cool project. So keep an eye out for when that gets released. And we hope you guys enjoy it. Then on to the next one, please. The next thing is I just wanted to make you all aware that COIN is now offering ClinVar submission brokering. So one of our main goals is to support variant sharing. And so if you're a lab who has variants for submission, but maybe you don't have the time or the resources to do the submissions yourself, you can do this through COIN. So if you're interested, you can just give the QR code in the bottom of the slide a scan, and you can contact us through our website. There's also lots of other fun things to look at on our website, but if you want to contact us for that, there's a form you can fill out for that. Then next is that we want to do a little teaser announcement, I suppose, for an event that we're going to be hosting at the end of the year. It's going to be a collaborative genomic event, and we are going to be inviting people to submit requests to be either part of an analysis team or cases for analysis. So if you are interested in participating or potentially leading an analysis team, just keep your eyes open and your ears to the ground, because we will be announcing more of that information soon. So best place to find that information is on our socials. If you scan the link on this slide, it'll take you through to our LinkedIn, which is where we post all of our stuff first. So give us a follow there and stay tuned for more exciting announcements to come. Thanks, guys.
Oh, thank you so much. Thank you, ladies. Anyone with a question? If you are feeling shy, you're more than welcome to. Okay, good. Claire, you're the star of the show. You can unmute yourself, Kuhle.
Hello, can you hear me?
Yes.
Yes.
Oh, okay, awesome. Thank you. I think that was a great presentation, and I'm basically going to direct my question to Melissa. So I just wanted to kind of ask what tools you use to assemble the genomes, and furthermore, like in the future, are you thinking of maybe incorporating your ultra long data to try to maybe access more regions that would not be accessible with just the long read data?
Yes. So to answer the first question, in our bioinformatics pipeline for long reads, we are aligning to the GRCH38 reference genome. So that really forms the backbone of our alignment and variant calling. However, in parallel, we are also building de novo human genome assemblies, which was another capability of the long reads that I didn't highlight specifically. But basically, because they are long, you can actually do a reference-free genome alignment. And we are interested in this whole other arm, which is centered around de novo assemblies and pangenomics, given the underrepresentation of our African ancestry research participants. And so, although we haven't quantified it specifically, we are interested in evaluating to what extent reference genome bias plays a role in our analysis and interpretation. So this means if you have a research participant who gives DNA to be sequenced, and the backbone of your analysis relies on aligning those reads to a human genome reference, which happens to be very different from the research participant, then there is a risk that the mapping will be misguided, or that there might be errors that are introduced. So in parallel, we are exploring de novo assembly and pangenomics, and perhaps we'll get to ultra long reads at some point, but not at the moment.
Thank you, Mel. Anyone else? I think there is a question on our Q&A. So one question is, what type of genetic testing are currently available in South Africa? How accessible are they? I don't know who it's directed to, Mel or Nebs.
So I can try and answer that. So I think when you speak about genetic testing, I mean, obviously that is a broad umbrella, because typically the tests that are offered are specific to certain conditions. So what we do know, if you look at inherited neuromuscular disorders, is that the local portfolio is limited. And we say that by virtue of the number of individuals who are able to achieve their genetic diagnosis in country. So although there are some tests available, it is not enough to diagnose everybody in whom a genetic diagnosis is currently possible. There are also obviously differences between the state testing avenues versus private. And so what we have been exposed to on the research front, being collaborating with clinicians at Groote Schuur, is obviously the public testing pathways. And so everybody who gets enrolled for whole exome sequencing has already exhausted the locally available diagnostic options.
I hope that answers your questions, Marai. Anyone else so that we can close? We're right on time so that we can. Okay, I think there's another question there. Okay. There's one more. So from Dr. Satih or Dr. Dumelo, great presentation. Are you using AI tools for your bioinformatics and variant interpretations? If so, how has your experience been with the tools?
So it's been revolutionary. We are using AI within proprietary software platforms to perform the tertiary analysis. So all the kind of up until secondary, which is taking the raw data to generate the variants, so the process of aligning it to the reference genome and calling the variants, that all takes place in our bioinformatics workflows on Elifu. And then once we have the variants, we port them into these proprietary software platforms where we leverage AI extensively to conduct the analysis. And it's really made it a lot quicker than it was before. So instead of doing, manual searches through gene lists, a lot of that process is automated. And we include the phenotype information alongside the genetic information. And the AI is really good at highlighting possible links between variants and phenotypes. So, yeah, to answer your question, it has really made things a lot faster. I think what is lacking to touch on that in the long read space is that although we now technically get all the variants, we are not necessarily solving cases just on account of having the full genome search space available. And I think what is lacking in that regard is our ability to interpret those variants adequately. So, using the right frameworks to interpret non-coding variants, perhaps discover new disease genes. And so I think AI is going to have an incredible role in helping us to really understand the variants that we've detected through HiFi sequencing. And we are actively tracking the developments in the field to see how we can really push our data to the limit and try and get more answers.
I'll just add on for the variant interpretation. I see a lot of international using VARChat and they're really praising the system. But just to bring it back to the underrepresentation of African data, and these models are trained on predominantly European data. So when it comes to interpreting the genetic variants, we're not yet there, and we do need human expertise and human experience to put the family history and the clinical symptoms and look at the bigger picture together with the variant that we identified.
Sorry about that. Thank you. We have another question from Mrs. Jacobs. Any future plans for interrogating unresolved cases or structural complexity with optical genome mapping?
Yes, so we have been exposed to that technology through one of our international collaborators. And it does seem to be an important adjunct or additional avenue to pursue for very large scale rearrangements. It's not something we've done yet, but I would say it's definitely a possibility for the future.
Right. I think that was our last question. We can take one more. Going once, going twice. Closed. Closed. I think there's another one that just came in. Oh, someone saying thank you for the great presentation. Great news about the Clinivar support that is highly relevant. It's from Dr. Lisa. Thank you so much, ladies.
Thank you for having us.
Well, I think I can close it. Where's my face? I promise I'm still here. Well, thank you so much. Thank you to everyone that took the time. I know it's after working hours for some of us, but thank you for honoring the invite as well and showing up for both us, RDSA, and COIN as well. Once again, Dr. Anil, Nabila, and Erin, thank you so much for being patient with me. Hopefully we'll do another great webinar soon. Maybe then we'll have at least 200 attendees or 200 registrations on board. No pressure. But thank you so much. Have a lovely evening further and enjoy the rest of your weekend. Okay.
Thank you. Thanks to everyone for listening.
Thank you.